Okay, so this lecture is going to start with acids and bases, and then we're going to work our way through macromolecules and talk about the 4 different macromolecules as well. Alright, so acids and bases, if you remember when we talked about water, how it will readily dissociate into hydrogen ions and hydroxide ions. Well, the way we measure acids and bases is through the pH scale. And the pH, what it's really measuring is the number of those hydrogen ions, or the concentration of those hydrogen ions in a solution. And so this is measured on a logarithmic scale. So I'll show you the equation in a second. But water is neutral, meaning it has the same number of hydrogen ions as hydroxide ions, or OH ions, and that occurs at a pH of 7. What that means is the concentration of hydrogen ions is 10 to the negative 7 mole per liter. I'll talk about that in a second as well. Compounds that are acidic are ones that have a higher concentration of hydrogen ions, and they're going to have a lower pH. Whereas basic compounds or basic solutions have a pH which is greater than 7 and a lower concentration of hydroxide ions. Now, one of the great things about water is that it is a buffer, and there are other types of solutions or compounds that when you mix with water make it a better buffer. And what a buffer is it is a solution that resists a change in pH. And so this graph shows then on the left here you have the pH scale, and on the right here you have the amount of this base added. So this is just a generic one, but this green line would then be the change in pH as we add this base. And as you can see on the tails here, on the left and on the right, you have a dramatic change in pH when you aren't really changing that much of the base. Right? Okay, so in the beginning when you start to add this base you get a dramatic change. But here in the middle, from about 1 to 4 times the concentration, the pH doesn't really change. And so this range here is called the buffering range. And basically what buffering means then is as you add more of an acid or a base, the pH doesn't change that much. So when an acid is added to this solution, or in this case a base, it absorbs the hydroxide ions. And when a base is added, it releases the hydroxide ions. And this range then, where that buffering occurs, is called the buffering range. Now outside of that, it is not a good buffer. So it's only a good buffer for a certain range. So the way we calculate pH is by taking the negative of the log of the concentration of hydrogen ions. So that's what all this means. pH equals the negative log, and these brackets mean concentration of the hydrogen ion. And it's measured in moles per liter, and a mole is 6.022 times 10 to the 23rd atoms. And you'll learn about that in chemistry if you haven't already. And so a pH of 7 means there are 10 to the negative 7 moles per liter of hydrogen ions in a solution. And so a pH of 6 means 10 to the negative 6. pH of 1 means 10 to the negative 1, so on and so forth. So why is this important? Well, when a solution has a very high or low pH, has a lot of those ions, which makes it highly reactive and also very corrosive, so able to start to corrode metals, or it is able to dissolve a substance very readily. And so bleach and ammonia are common cleaning products which are used because they are on the extremes of the basic scale, and thus they will help remove dirt and grime and other things because of that very low pH or high pH. Hydrochloric acid then has a low pH. It's an acid, and it is a principal component of what's found in your stomach, and so that is used then to break down and activate enzymes. So used for digestion. All right, so now we're switching to macromolecules. Now, a very important thing about making macromolecules is you need to have you need to be able to chain things together. And what makes this possible is the structure of carbon. So carbon has 4 outer electrons on its valence shell. So it can make 4 covalent bonds to fulfill its octet. That means it can bind to 4 different things. And it also readily bonds with oxygen, phosphorus, sulfur, nitrogen, and hydrogen. And these are plentiful elements found in living things. So a hydrocarbon then is a chain of carbons with lots of hydrogens on them as well. So ethane, propane, and butane are repeating units of methane, and that's what hydrocarbons are. These are small hydrocarbons. They get much larger when we start making things like plastics. So another chemical component which also can affect a compound and its reactivity is this principle of having isomers. And these have the same molecular formula, but they differ in their structure. That's what a structural isomer is. So you can see these two things are actually the same. They have the same components. These are just generic letters to represent different elements, but they aren't exactly the same because they are opposites in their configuration, just like your right and left hand are. They're pretty much the same, and they are mirror images of each other, but your right hand is not exactly the same as the left hand. No matter how you spin over either of your hands, you can't make them exactly the same. You can also have ones that are different based on attachment, and those are stereoisomers. And enantiomers are then molecules that are mirror images of each other. So this is an enantiomer example over here. Those that are said to have this type of arrangement are called chiral molecules. Alright, so carbon then is used to build macromolecules. Macromolecules are all polymers, which means they are a large chain of a repeating smaller unit called a monomer. Polymers have bonds which are broken by hydrolysis. And so what happens is we have this molecule, let's say, of 3 carbons here. In order to break this bond and for it to be stable, you have to have a hydrogen on one side and a hydroxide ion on the other. You don't have to, you could be another hydrogen, but because water is generally abundant, you can split a water and it will act very readily on that carbon molecule and make 2 stable configurations. So that's called a hydrolysis reaction, because it takes the water, takes the hydrogen from one and the OH from the other part, and makes 2 new molecules when that bond is broken. The opposite occurs when you are making a larger molecule. That's called a dehydration synthesis. So if these 2 carbons are going to bind together, you need to get rid of the H and the OH and form a water molecule. And so that's what happens when you form a larger molecule from 2 smaller molecules with an H and an OH on them. So the 4 macromolecules of life are the carbohydrates, nucleic acids, proteins, and lipids. Carbohydrates typically have a 1 to 2 to 1 carbon hydrogen to oxygen ratio. And the bonds between the carbon molecules store energy, and in plants, when those molecules are made, it's called starch. Animals make a form of sugar molecules combined together called glycogen. A monosaccharide, saccharide refers to sugar, so a monosaccharide means it's one sugar. And these are 6 carbon sugars. Examples of those are fructose, glucose, and a galactose. Now these have the same chemical formula, but are arranged somewhat differently. So fructose and glucose are structural isomers. Glucose and galactose are stereoisomers. They are the same, except the OH and the H on the 1, 2, 3, 4th carbon is flipped. Whereas fructose and glucose are the same, except the placing of the double bonded oxygen is on the 1st carbon and the 2nd carbon. A disaccharide, di meaning 2, is 2 sugars which are bound together. Examples of those include lactose, sucrose, maltose. And then you have polysaccharides, which are 3 or more monosaccharides combined, which include cellulose, lignin, and chitin. The monomers then of these saccharides, of these sugars, of these polysaccharides are sugars, monosaccharides. Okay. Alright, our second one is nucleic acid, and this is a polymer of nucleotides. Okay, so the monomer then is our nucleotide. A nucleotide is made up of a phosphate, so that's what this is right here, which is a phosphorus surrounded by oxygens. A sugar molecule, here well, I'll talk about the different sugars in a second, and then a nitrogenous base. In DNA, deoxyribose, nucleic acid, the sugar is deoxyribose. Okay? In RNA, the sugar is ribose, ribonucleic acid. The only difference between DNA and RNA is at this 1 carbon, so this represents a bunch of carbons strung together. There's a hydrogen in DNA and a hydroxide in RNA. The bases that are used are slightly different as well. In DNA, you have adenine, thymine, guanine, and cytosine, and in RNA you have the same, except you don't have thymine, you have uracil. And these nucleotides are bound together by a bond called a phosphodiester bond. And so that's what you have here. Phosphodiester bond binds this nucleotide to this nucleotide. So DNA then, of course, is one of our examples of a nucleic acid, and it has adenine, guanine, cytosine, and thymine. That's what those stand for. It's arranged in a double helix, so kind of like a twisted ladder, and the bonds along the backbone, which we mentioned before, are phosphodiester bonds, but across the bases also form hydrogen bonds between them. Ribonucleic acid is a single strand of nucleic acids strung together, so it doesn't make the twisted ladder of DNA. And there are 3 different types. You have messenger RNA, which is formed at the DNA, so it's read during a process called transcription. We'll go into that in a later chapter. rRNA, or ribosomal RNA, is what ribosomes are made of. And tRNA, or transfer RNA, takes amino acids to the ribosome as they're being read by the messenger RNA, and the ribosome then strings those amino acids together to form proteins. So some other types of nucleotides are ATP, adenine triphosphate, and this is the ubiquitous energy molecule. NADH and FADH2 are also used to make energy in the electron transport chain, which we will also talk about at another chapter. Alright, our third macromolecule is proteins. And these are polymers of amino acids, which we just talked about with the RNA. They have lots of different functions. In fact, the proteins are basically the workers of the cell or of the body. So they are important in helping reactions move faster in the form of enzymes. They can help defend against invading pathogens. They help to transport hydrophobic substances through the bloodstream. They are there for support, so your muscles are made out of proteins. They are also important for motion, right? That's your muscles contracting is how you move. They help regulate the influx and outflux of things going in and out of cells, and they are also important for storage. Okay. So here we have. We'll go over what this is, but this is where DNA is made. Okay, so which is nucleic acids, but what is going on is you have a bunch of different enzymes which are actually doing all the work and putting the strands of DNA together. Okay, this then is a muscle cell, which is different proteins called actin and myosin strung together to form these fibers of muscles. Okay, and then you also have proteins which are embedded into cell membranes, which allow substances in and out. And finally, catalase is an enzyme which helps break down hydrogen peroxide into, which is a very reactive form, to a less reactive form of water and oxygen. So amino acids are bound together by what's called a peptide bond. Okay, and this is going to involve two amino acids and then a dehydration synthesis to make a dipeptide or two peptides. Every amino acid has a central carbon which attaches to a hydrogen on one end, a carboxyl group on one end, an amino group on another end, and all of those stay the same on every amino acid. All of them have these. But the R group is different. So it can be as little as just an H, or it could be a whole string of carbons and with tails and everything on it. So the R group is what defines the type of amino acid it is. And like I mentioned before, the peptide bond is formed by a dehydration synthesis between the carboxyl group on one end and the amino group, which is the NH2 group, on the other end. And these are some of the amino acids. There are 20 different amino acids, and they all have different properties. Some are charged, some are polar, nonpolar, some are aromatic, meaning they are detected by our smell very easily, and some are complex and have a very special function. Those are all the amino acids. Now, there's different levels of structure when you start putting these together. The primary structure is just the sequence of amino acids. So you may have a protein that's 4 amino acids long, and it's made of 2 phenylalanides, then a tryptophan, and then a lysine. Okay, so that's the primary structure. The secondary structure is after you have the primary structure done, then you have interactions between the R groups, okay? And they form different structures. One of them may be a alpha helix, which is what we have here, or a beta pleated sheet, which is what you have there. The tertiary structure then is the final folded shape of the chain of amino acids. So after you have all of these secondary structures combined together, it makes this larger chain of amino acids. And then you may also have a quaternary structure where you have multiple tertiary structures combined together to make a larger protein. So an example of this is hemoglobin is made of 4 interacting globin subunits. Finally, after a ribosome makes a string of amino acids, it still has to be folded correctly in order for it to function. So it does this with the aid of what are called chaperones. Okay, one example is in the disorder called cystic fibrosis. These proteins are not folded correctly, and so they do not function. So this is referring back to one of our themes in biology, which is that structure is related to function. If it's not structurally sound, then the function will not be there. Another thing that can happen if a protein becomes too hot or too cold, those bonds that form between the R groups and the secondary and tertiary structures can start to break and warp, and then that will also cause the function to be altered as well. Alright, our last macromolecule are lipids. These are defined as nonpolar hydrophobic molecules and include fats, waxes, oils, and some vitamins. So some examples here. Gasoline is a hydrocarbon, so it's a long chain of carbons and is a nonpolar substance. Leaves, I have 2 leaves here. Leaves produce a waxy layer on top of them called a cuticle. And so that wax then is polar. You may also have the production of lipids by fruits or nuts. So olives create this oil, which is we use in cooking. And then you have some vitamins. So this is vitamin A, which is synthesized by many plants and a nutrient necessary for life. And so when you eat food, you get vitamin A from your foods. Now, there are different types of lipids or different arrangements. Well, one of them are fats, which are made of a glycerol backbone. So that's this kind of 3 red molecules stuck together there, attached to fatty acids. So these are 3 fatty acids and a glycerol. So that's a triglyceride that makes a fat. Some of them differ in structure depending on the double bonds in their fatty acids. Okay, so you see in here you have, between these 2 carbons, you have a double bond. And what that does is it causes those 2 carbons to kink. When they have single bonds, they're all kind of flat and packed together. And when things are flat and packed together, they start to form bonds between them. When they are kinked and far away, then they don't form bonds between them. And so the ones that are packed together are more solid. And so these are fats that are at liquid or room temperature are unsaturated fats. Sorry, these are saturated fats, meaning they're saturated with single bonds. Whereas unsaturated fats have these double bonds, and these are usually liquid at room temperature. Okay. For the most part, the lipids produced by plants are oils and are liquid at room temperature. But some of them can be solid, such as in avocados, which are filled with fat. Alright, finally, lipids can also be used to make phospholipids. Okay, there are 3 parts of the structure of a phospholipid. You have a hydrophilic phosphorus head attached to a glycerol molecule, which is then attached to a lipid tail, 2 lipid tails. Okay. and that makes our phospholipid. These are very important for making the phospholipid bilayer. So that's what we have here. Okay, which makes up which is the main structural component in cell membranes. All right.